Okay, everyone, welcome back from the poster session. Now, continuing, I'm here to introduce our next speaker. My name is Vipul Kumar. I am a graduate student here at Iowa State University. I'm working with Dr. Fay in the Department of Horticulture. So, I'm going to introduce our next speaker for today's symposium. Boris Alladassi is a PhD student in plant breeding with a statistics minor in Dr. Janming Yu's lab here at Iowa State University. Boris' PhD research focuses on integrating phenomics, genomics, and transcriptomics for complex trait dissection. He has been involved in the leadership of several student organizations. He served on the organizing committee for the R.F. Baker Plant Breeding Symposium for 4 years, including a term as co-chair in 2022. Currently, he is the college chair for the College of Agriculture and Life Sciences at the Graduate and Professional Student Senate. Today, he will be presenting on the genetics of leaf angle across the canopy of maize. Please join me in welcoming Mr. Boris Alladassi.
Thank you, Vipul, for the introduction. Good morning, everyone. First, I would like to say thank you to the organizing committee for giving me this opportunity to present my research. And this morning, I will be talking about the genetics of leaf angle. So I would like to start with three fundamental concepts in plant biology. On the left, we have ontogeny that deals with the development of the organism from its conception through adulthood. In the middle, we have allometry that deals with the differential growth of different parts of the organism throughout development. And finally, we have plasticity that deals with the changes in the phenotype of the organism across environments. And there have been several long-standing questions about the genetic basis of these three different aspects of development. And now that we have a wide array and we can able to integrate this data all the way from the genome to the phenome as well as the environment, we're able to answer some of these questions, but also ask new questions. So my research in Dr. Yu's lab deals with primarily the first two aspects of development. First, my project one is leveraging the spatial and temporal resolution of phenomics combined with genomic technology to investigate the genetic basis of ontogenic growth in sorghum. I'm not going to talk about that project this morning. I'm going to talk about the next one, where we are using genomic and transcriptomic data to dissect the differential control of leaf angle across the canopy of maize. So on the first slide, we were looking across time, but on this one, we are looking vertically. But let's start with why leaf angle. I'm sure many of you have already seen the graph on the left as on several occasions. So we can see that there have been a remarkable yield increase for maize in the US over the last 80 years. And studies have shown that increase in plant density from 30,000 to about 80,000 plants per hectare is a major contributing factor. But at the same time, there have been also genetic improvement. These cultivars underwent some genetic changes that allow them to be adapted to the changes in density. So from a shorter anthesis silking interval to a smaller tassel, we also have an increase in leaf angle. So we can see here on the left, in an error study published by Duvik et al. In 2004, how leaf angle score increased from 1 to 9 over time. So modern cultivars have more upright leaves compared to their older counterparts. But this study, as we can see, is using a leaf angle score, which is kind of an average representing the whole canopy. But we know there is a natural variation in leaf angle pattern across the canopy. Here we are showing three inbreds here. We have Mo 17, a non-safe stock inbred, where we have generally horizontally oriented leaves. But in the middle, we have B73, which has a dynamic leaf angle where the leaves are becoming more upright as we go to the upper canopy. And finally, we have PHW30 and iodent XPVP, where we have generally upright leaves throughout the entire canopy. So given the importance of leaf angle in the adaptation to increase plant height, where we are able to get with the upright leaves a deeper penetration of solar radiation within the canopy, we think it is also important to ask another question instead of just focusing on, let's say, an average of the canopy or looking at only one leaf of the whole canopy. So we are interested in how is the genetic regulation of leaf angle across different layers within the canopy. And then the second question that we are asking here is about what is also the optimum canopy for grain yield in maize. And to answer this question, our lab developed a set of double haploid lines that we use. So first we have B73 that was crossed to PHW30, both forward and reverse. And we also have MO17 that was crossed to PHW30 forward and reverse. And we generated 309 double haploid lines. So we evaluated these DH lines for 3 years in the field. Yes. And the primary trait that we collected data on was leaf angle. And since we are interested in looking at the variation across the canopy, then we measure leaf angle at 4 points, representing spanning throughout the canopy. So first, I would like to mention that leaf angle in this case is the inclination between the midrib relative to the horizontal plane. So higher value means more upright leaves, while lower value means flatter leaves. So we measure the leaf angle at the second leaf below the ear, the first leaf above the ear, third leaf, and then the pre-flood leaf at the end. And we analyze the data in R. As you can see here, thanks to the undergrad that was helping me that day, that's me measuring LA-2 in the field. Now, let's go to the result. First, we look at the correlation of leaf angle across these different canopy positions. And what we see is that there is a positive correlation from moderate to strong correlation. And as expected, the correlation between adjacent canopy positions are stronger than when we look at canopy positions that are further apart. First, thus confirming that we have some dynamic leaf angle for some of the DH lines. But then to characterize that further, we did a cluster analysis to see how they are grouping according to the leaf angle pattern across canopy. And here I'll be showing from the right to the left, we have first cluster 1, where we can see that it has upright leaves, so higher leaf angle throughout the canopy. But as we go to the second cluster, we see that the leaf angle increase as we move from the lower canopy to the upper canopy. The next one has mostly for cluster 3, we have upright leaves, but then at the top of the canopy is a little bit more open. And we have the next one, which is more intermediate leaf angle across the canopy. And let's go to the last one, which is cluster 6. After carefully looking at it, we actually found that there are 2 types within that cluster. The first one, which has overall lower leaf angle, so mean horizontally oriented leaves across the canopy, but also an interesting one, where we have somewhat upright leaves at the bottom of the canopy, but then flat leaves at the top of the canopy. And that was kind of interesting to see, which is unique. Now, from here, we now go to the QTL mapping. And again, our interest is to look at the variation across canopy. So we did the analysis in 2 stages. First, we analyzed each canopy position at a time. And then since we already saw that there's a correlation between this trait, we also ran a multivariate QTL analysis. So the result that I'm showing here on the left, we can see for the dots, the circle, those are QTLs that were first detected from the univariate analysis and then further confirmed from the multivariate analysis. While those that we are showing here with the square QTL, additional QTL that we detected exclusively from the multivariate analysis. And looking at the pattern, what we see, for example, the one in pink, we can see that this QTL was detected for all 4 canopy positions. While the subsequent QTLs, still in the circle, they affected 2 to 3 different canopy positions within the canopy. And finally, we have this QTL on chromosome 7 that was only detected for the upper canopy. So this is interesting. We zoom into the region to see if there are some underlying candidate genes. And we found 3 of them, especially ligulus 3 and ligulus 2 genes that are known leaf angle genes in maize. But these other ones, we are also still interested in investigating further, the other ones. Now, let's look at how are these QTL, the effects across canopy position. So we have multiple patterns, but here I'm showing 4 representative ones. So we can see, for example, for the upper left one, we can see that the QTL is affecting leaf angle across the entire canopy. But next to it, on the right side, we can see that we are seeing a type of crossover pattern, which is interesting. Now, the lower left one, we can see that the QTL is detected for only the lower 2 canopy position. While for the last one, we can see that it's affecting only the upper canopy position. Now, in addition to this result, we've conducted also some transcriptomic analysis. And from the result that we have so far, we are seeing some interesting enrichment terms such as photosynthesis, as well as some hormonal signaling. And there are some differential expression pattern, especially for leaf angle genes. But for the sake of time, we are not presenting all that today. So from here, we can now go to the second question that we asked about the optimum canopy leaf angle pattern for grain yield. So to answer that question, we now cross the 2DH population, the 2DH population to the 3 inbreds to develop 912 hybrids that are shown here in this 6 combination. And we evaluated these hybrids across 3 environments here in Iowa for 2 years. And we planted them at a population density of 79,000 plants per hectare. The primary traits that we collected data on were leaf angle and grain yield, but also other potential traits that can affect grain yield that we fitted in the model as covariate to adjust for the effect. Now, the first point we were interested in to see how consistent is the leaf angle pattern between the DH line and their respective hybrids. So looking at these 4 panels representing from the bottom left here, which is the lowest canopy position to the upper canopy position, we see overall that there is a consistent leaf angle pattern between the DH lines and their respective hybrids. But to the more interesting question about the correlation, well, it turned out there was no linear relationship between the grain yield and the leaf angle at first. And that was like, as a graduate student, I was looking for that correlation and then, ooh, no correlation. Okay. Instead, what we are seeing is kind of a non-linear pattern in the data. So we decided, okay, what is the underlying pattern yielding this? So we decided to look at the genetic structure of the population. How did we look at it? First, we look at the 3 different test cross populations that we have. Now, when we look at the correlation within each population now, we can see that on the left side, we have the hybrid from the Mo17 tester, where the cloud is on the left, so lower leaf angle, meaning more flat leaves. We see that higher leaf angle is associated with higher grain yields in this case. And if we look at the other 2, where we have the parents, the testers have mostly upright leaves, we are seeing a negative trend. So that is what was creating that pattern that was seen earlier. Now, from the test cross population, we zoom in again and now consider all 6 combinations. And from the 6 combinations, again, we see lower leaf angle, there is a positive association with grain yield. But as leaf angle increases and the leaves become too upright, when we start seeing a negative correlation. And there is, in the literature, we found Lambert and Johnson in 1972 already reported this, whereby if the leaf become too upright, the solar radiation is hitting mostly the abaxial surface of the leaf instead of the adaxial surface. And therefore, that may not be beneficial for photosynthesis. Now, there are two other combinations that are very interesting. And these two combinations here. The first one is here on the left, where we have the DH with the B73 background cross to MO17. And we know these two heterotic group. And then the second one is this one in the lower corner here, where we have the MO17 DH background cross to B73, two different heterotic group again. For those one, we can see they are above, they have a higher yield, but then there is no correlation between grain yield and leaf angle for those two combinations. So we decided to go in and see how are they grouping depending on the clusters that we looked at before. So we already know that there's a consistency between the leaf angle pattern between the DH line and the hybrid. So if we look at the 6 clusters, how are the hybrids that were developed from those DH line, how are they yielding? Well, we see that for cluster, for the one on the left, overall cluster 4, where we have intermediate leaf angle across the canopy. And then for cluster 3, where we have upright leaves, but the upright is not too upright, but open at the top, those seems to have grain yield. For the second, there is no a significant difference, but still for cluster 4, we can see it tends to have a higher yield. So these are some of the questions that we've tried to answer, and we can now summarize. First, we were able to characterize the leaf angle pattern across the canopy of this DH line into 6 different clusters, and then we detected 8 QTLs, some of which are having stable effects across the canopy pattern, while others are having a dynamic effect across the canopy pattern. And from the correlation analysis that we did, we can see that, well, increase in leaf angle from flat leaves is associated with higher yield, but if the leaves become too upright, it may not be beneficial in the end. On that, I would like to thank my lab members, especially Dr. Jamin Yu, my advisor, for his incredible support, and other lab members whom we always have very engaging discussion during lab meeting, but as well, previous lab members that have really paved the way for us to do what we are doing today, and also the funding agencies. On that, thank you very much, and I'll be happy to take any question.
Thank you, Boris, for the presentation. Now I would like to reach out to the audience if someone has.
Boris, thank you for the presentation. Just a quick question. Did you look at the amount of variance explained by your QTLs?
Yeah, thank you, Radu, for that question. Yes, we look at the variance explained by the QTL. And for these 8 QTL that we presented, they explain about 29% of the variation in leaf angle. Thank you.
Yeah, really nice talk. This is Somag Datta. I have a couple of questions. One is, did you measure the leaf angles in the field trial? Did you measure the leaf angles again at the 4 different heights? And the second, okay.
Yes, yeah, although I didn't show that, yeah, for the yield trial of the hybrids, yeah, we measured the leaf angle again at 4 canopy position, just like we did for the DH lines.
And did you look at, instead of a correlation between the average leaf angle and the grain yield, did you look at correlation between the grain yield and leaf angle at every individual heights, or multiple kind of linear regression model on the granules on those?
Yes, that's an excellent question. And yes, we did that, although given the time, we couldn't present all that detail here. Yeah, and what we presented here was the average across the 4 canopy position. And the trend is very similar to what we saw for each individual canopy position as well. Yeah.
What's the heterotic group that PW line is?
Pardon?
What's the heterotic group that the PW line that you used as a parent?
Oh, yes. Yeah. PHW30, yeah, is an iodine, XPVP.
Have you taken a look when you made the cross, you know, the MOS 17 and the B73 with that PHW? Did you take a look in the DH as to where the DHs would cluster on which side of the heterotic group? Because with some of the information that is coming, would that be an artifact, or maybe there's some more information that may come based on, you know, how they combine with that particular tester that you ended up using?
Yeah. Yeah. So in this case, we crossed B73 to PHW30 for the first population, and then we crossed MOS 17 with PHW30. Yeah. So the gauge population didn't have B73 by MOS 17. But look, because we know B73 is from the stiff stock, and PHW30 is the iodine. The trend that we are seeing the relationship between grain yield and leaf angle. Yes, the heterotic group, when combined, we are seeing a boost in yield, and that's why we went ahead and looked at those 2 specific combinations to see, even within that particular combination, how is the grain yield relating if we look at different leaf angle patterns. And that's where we saw that for canopy architecture 4, where we have overall intermediate leaf angle, we are seeing higher yield. Now for the other combination as well, as we look, we also have for the cluster 4 as well, especially for those where we are seeing a negative trend. And for the one that we are seeing a positive trend is about for cluster 3 and cluster 2.
I see your variation across the year for the leaf angle very dramatically different. And that increase associated with the yield. I wonder, in the current cultivar or current germplasm, the breeding germplasm, how big the variation, how big a potential those variation can further improve the yield?
Yeah, that's a good question. And it was one of the interesting things that we are trying to see from this data. Right now, I think the cultivars that we have, they have overall upright leaves throughout the canopy. But we know that there was the concept of maize isotype that was proposed in 1975, where if we could have more horizontally oriented leaves at the bottom of the canopy and upright leaves up of the canopy. And I think we can still be able to test. So once we identify these genes that are having differential control for leaf angle, we can have different combination to produce different canopy type under the same background. And then we can test exactly, in more detail, how the canopy changes is affecting grain yield.
Okay. The following question is, I'm alfalfa breeder. We breeding for the herbage, and we're looking for the leaf angle, because if you have more herbage yield, definitely we have higher biomass yield. We directly harvest the herbage. Well, for sorghum or other crops, we harvest the grain. Is there a relationship? You have higher synthetic with angle that can absorb more light, and then you increase the biomass yield and also the grain yield. How much of those energy from bio energy portion to the grain portion, whether the angle, the gene control angle with the relation of the gene control angle with the gene control of the grain yield.
Do you mind Would you mind coming again? Okay. I couldn't follow all throughout, yeah.
I was going to suggest that too. Thank you for the lively discussion, but due to time, thank you very much, Boris, for your great talk.
Thank you.
Now, Priyanka Tamineni will introduce our next speaker.